Welcome to our discussion of meiosis. This is the fourth presentation of 4 presentations for Science 30, Unit 2, the Biology Unit. Mutations occur when there are changes in DNA. Mutations are defined as heritable changes in DNA, and they arise from mistakes in DNA replication. They change the genetic code of the organism. Incorrect DNA builds the wrong protein using the wrong amino acid. AA stands for amino acid. Most produce adverse effects, but can also be a source of genetic variability. So in this case, you have a mutation here where the protein should have said CGC, but now it says, or GCG, but now it says GAG. So this is what we call a point mutation. And instead of coding for a triangle, it codes for a stop, so it prematurely ends your protein. So here's an example of a the results of a mutagenic agent. Mutagenic agents cause mutations. They include radiation, so X-rays, UV radiation, and microwave radiation. They also include chemicals. For example, chemicals in cigarette smoke. Also, the hepatitis virus can cause mutations in the immune system. Mutations in DNA, they can be chromosomal, so changes in the structure or number of chromosomes. Here we have, instead of 2 chromosome 21s, we have 1, 2, 3 chromosome 21s. They can also be a point mutation or changes in the part of the DNA. So instead of this being cytosine, thymine, thymine, it is now instead thymine, adenine, or cytosine, adenine, thymine. So this is a point mutation where we're changing T for A, and which results in coding for instead of glutamine, you have valine or whatever. Changes in the structure or number of chromosomes. So normally in humans, the number of chromosomes we should have are 46 or 23 pairs. 46 is known as the diploid number, or the 2n number. How do we know whether or not people have more chromosomes? Well, we need to do something called karyotyping. Karyotyping is the organizing of chromosomes into a chart according to 3 different characteristics. They arrange based on the size, and then when you stain it, you can get colors, and then they arrange it from the banding pattern, so similar banding patterns, and centromere position, so middle, top, bottom, things like that. This is used for detecting chromosomal abnormalities or mutations. Here we have the sex chromosomes. We have an X chromosome and a Y chromosome, so this individual is a male. Autosomes are considered to be chromosomes 1 to 22. They're not involved in gender determination. The sex chromosomes are the 23rd pair of chromosomes, and they can be X or Y. They determine gender. Females are XX or XX, and males are XY. Karyotyping produces an organized chart of the chromosomes in pairs. So when you karyotype something, or when you start karyotyping something, you get this jumbled mass of chromosomes, and then they take imaging software and they cut and they paste these into these pairs of chromosomes. What is abnormal meiosis? So sometimes you get something called non-disjunction. And this occurs when homologous chromosomes fail to separate and move from the same pole during the anaphase of meiosis. Abnormal gametes with abnormal numbers of chromosomes form things like trisomy 21, which is Down syndrome. Genetic disorders result. Chromosomal abnormalities, so looking at non-disjunction in meiosis 1. So here you have metaphase 1 occurring normally, and then in anaphase 1, instead of this pair going this way and these two going this way and these two going this way, you have all three moving up and only one moving down. So this is called a trisomy 1, 2, 3. So here we have a 2n cell resulting in an n plus 1 cell, n plus 1 cell, and then you have n minus 1, n minus 1, meaning that these two have one more chromosome than they should, and these two have one less chromosome than they should. Down syndrome is this condition called trisomy 21, which means that people with Down syndrome have 3 chromosome 21s instead of 2 chromosome 21s. It's also known as trisomy 21, which means, again, 3 pairs of chromosome 21. About 1 in 600 babies have Down syndrome. The incidence of Down syndrome increase with maternal age. So as women age, their chances of having a child with Down syndrome increase. Turner syndrome is a condition where people have one less sex chromosome than they should. So they have an X chromosome but no second X or no Y chromosome. They're considered to be female because they show female characteristics. It is again due to non-disjunction during egg formation. One egg gets both X chromosomes and the other egg gets no X chromosomes. So they're getting that X chromosome from the father. About 1 in 10,000 births exhibit Turner syndrome. Klinefelter syndrome is a non-disjunction event as well. It is a trisomy of your sex chromosome, so individuals have 2 X chromosomes and 1 Y chromosome. It is again due to non-disjunction of the sperm or the egg. They appear to be male at birth but are sterile and produce female hormones. So you get things like breast development and small testicles and not as much facial hair or body hair. About 1 in 10,000 births show symptoms of Klinefelter syndrome. Jacob's syndrome is another non-disjunction event. So they consider it to be like the super male because these individuals have 2 Y chromosomes. These people tend to be more aggressive than average males. How do people detect genetic disorders? One way is through ultrasound. It locates the position of the fetus. Another way is to take a sample of the amniotic fluid, and that's called amniocentesis, which uses a needle to withdraw some amniotic fluid from around the fetus. The fluid is in the uterus, and fetal cells can then be isolated and analyzed. Another way to do it is to come in another way through the vagina and do transcervical chorionic villus sampling. So again, it's another way to get fetal cells to isolate, but not to stick it through the mother's abdomen. It draws some cells from the outer membrane of the embryo. CVS can be used as early as 8 weeks of pregnancy. Point mutations can come in a variety of flavors. One is a frameshift mutation, so that's the addition or deletion of a DNA base. The other one is non-frameshift, so it's substituting one base for another. So in the case of what we're talking about, substituting adenine for thymine or cytosine for guanine or whatever. Point mutation substitutions. One point mutation that one common disorder that is the result of a point mutation is sickle cell anemia. So where one change in the DNA from cytosine thymine cytosine to cytosine adenine cytosine results from a change from glutamine to valine and results in these sickle cell red blood cells. When one or two bases have been substituted, point mutations or substitutions are when a base has been substituted for another accidentally. Only one triplet on the DNA is affected. Example is if guanine thymine changes to guanine cytosine adenine. The resultant mRNA or the instructions that make proteins change from CCA to CGU. Then the incorrect tRNA, or the part that attaches the amino acid to the protein, is attracted, and the wrong amino acid is placed in the protein or the polypeptide chain, resulting in an incorrect protein being formed. Sickle cell anemia was caused this way. Interestingly enough, they think sickle cell anemia it persisted in the population because the people with sickle cell blood cells had a greater resistance to malaria. Point mutations can also be deletions. There's often these have a more serious consequence because a base is accidentally dropped from the sequence. Many of the DNA triplets can be affected because the deletion alters the sequence or the way that the code is being read. So if you skip a letter, you affect everything downstream of that letter. It's like dropping a letter from a sentence. For example, sometimes the error may arise, becomes some est rorum aya rise. So you can see that it doesn't make sense. A base is added to the DNA strand in an addition. The triplet with the addition is affected, and so are the rest of the triplets afterwards. So downstream is affected as well. For example, if a DNA before mutation is AAA, TTT, CCC, GGG, TTT, CCC, after the addition of a new sequence, it would read AAA, TTT, and now you have an addition of a G, GCC, CGG, GTT, TCC. So you can see how everything gets shifted downstream.